Okay, hello everybody. I hope you can hear me well. So, welcome to the VirusDetect pipeline webinar by CSC. I'm Eija Korpelainen. Also, my colleague Kimmo Mattila will talk today, and Maria Lehtivaara will also be present, taking care of the technology and also answering questions. And finally, I would like to already at this stage say a big thanks to two people who are not from CSC. So these people, there is Jan Krois from the International Potato Research Institute in Peru, and Professor Jari Valkonen from the University of Helsinki. They have helped us a lot in setting up the VirusDetect pipeline at CSC. So what we are going to talk about today is, first of all, I give a very brief introduction to the background, so to the small interfering RNAs as an antiviral defense mechanism. And the main point is really this one. So we will be discussing the VirusDetect pipeline, the central concepts of it, the analysis steps that are performed, and we also look at the result files. Then I will introduce you to how to use this tool in the graphical Chipster user interface, which you can operate with mouse. And after me, Kimmo will tell how you can use VirusDetect on command line. So if you prefer, if you're comfortable working on command line, perhaps doing even some programming, then using command line on the Taito cluster might be the way for you. But let's start with the background first. So RNA interference, as you probably well know, is an antiviral defense mechanism in eukaryotic organisms. So basically, double-stranded RNA, the viral double-stranded RNA molecules are cut into small interfering RNAs or siRNA molecules, which are from 21 to 24 nucleotides long, and which then target this complex to degrade the viral RNA. And there are also biological mechanisms, which actually amplify this system. So as an end result, cells which have virus infection actually have a very large amount of these small interfering RNAs. And we take advantage of this in our virus detection system. So basically, we sequence the small interfering RNAs, and since they are pieces of the full virus, we assemble these RNAs into full virus sequences, which we can then compare to known virus sequences and thereby detect the viruses. So, so we extract RNA, size separate the molecules, take the fraction which contains the very short 20 to 30 nucleotide RNA molecules, and send them for sequencing. When the data comes then from those very short sequencing reads, we need to figure out what viruses are present. There would be several ways to do that, but one of the most recent methods is called VirusDetect, which was developed at the Cornell University in the US by Cheng et al. And essentially, it's a pipeline that combines several analysis tools in one tool, so it's very comfortable to run. So what it does, the main points are these two here. So it assembles this short RNA reads, small RNA reads, to longer sequences, which we call contigs, and it does it in two different ways to make sure that it catches everything. So one way is called reference-guided assembly, which means that the sequencing reads are aligned to known virus sequences, and then the matching pieces are combined together. The other approach is a de novo assembly approach, which means that no virus reference sequence is used, but instead only the reads are used. They are matched to each other, and the contigs are built from that. So essentially that's like doing a puzzle without the actual model picture. Now, once the contigs are available, then we go and compare them to known virus sequences using BLAST. So let's have a look at this in more detail. And as you can see, many of these figures are kindly provided by Dr. Jan Kroes from the International Potato Institute. So on this figure now, we have the reference guided assembly. So here we have a reference sequence, some virus sequence that is 1,000 bases long. And then we have our short sequencing reads, which are these little blue bits here. So what we do is we match the sequence of the read to the virus sequences, and then we look for overlapping reads like what we have here, because those we can combine together and construct these contigs. So this particular contig here is 600 bases long, and then we have another one here which is 200 bases long. So altogether we have managed to cover 800 bases of this particular virus. So we say the coverage is 80%. And you will see that we also will talk about the depth. So that means how many reads on average cover each position of the virus reference. Now, how this is done in practice is that those reads are aligned to the virus reference database using an aligner called PWA, which is quite a well-known aligner in the next generation sequencing analysis. And as a reference virus database, the VirusDetect pipeline uses GenBank virus sequences, which have been sort of pre-treated in that they have been first of all classified to 8 different host kingdoms, which you see here. So for example, if you are looking for potato viruses, you might not be interested in looking at vertebrate viruses, so you would concentrate on the plant subsection. Also, the VirusDetect people have removed redundancy. In other words, if there are sequences which are very similar to each other in the virus database, they have been combined. And this same sequence set is then later on used for the BLAST similarity searches. So during the reference guided assembly, the reads are aligned to the reference using PWA, and then the overlapping reads are sort of merged together using a tool called SAMtools. The other assembly then, the de novo assembly, is done using a program called Velvet. And before the Velvet tool is run, VirusDetect pipeline actually cleans the data a little bit. So it will remove those small RNAs which are coming from the host. So naturally, when you extract small RNAs from a cell, inevitably there will also be small RNAs which come from the host and which are not virus-derived. So those host-derived ones you want to get rid of, because imagine if you are making a puzzle and somebody throws in lots of pieces from another puzzle, your work would be much harder. So we want to get rid of the non-viral small RNAs, and we do that by aligning the reads first to the host genome with the PWA, and then we continue making the contigs with those reads that did not align to the host, because presumably they are the viral ones. Once Velvet has assembled the contigs, they are still cleaned just in case. So the ready-made contigs are still compared to the host genome, just to make sure that we are not actually analyzing host sequences, but the viral ones. So the This picture here shows what exactly happens in the assembly part of VirusDetect. It might not be very clear on your screen, but you find the same picture actually in the Chipster manual page, which I will show you later. So we start off with the small RNA reads, and we do the two different assemblies, the de novo assembly and the reference-guided assembly. The reference-guided assembly is very simple. Like I said, reads are aligned to the virus reference database and assembled to contigs. And on this side, before the de novo assembly, the reads are cleaned so that the host-derived small RNA reads are removed, and the rest is used for the assembly, and the contigs are also cleaned, and then the final set is combined with this other set obtained in a different way from the same reads, and the redundancies are removed, and you end up with your final set of contigs. So then for these contigs, we need to perform the actual virus identification. That is done with BLAST-N against the virus reference database that I just described earlier. And the BLAST-N results are reported. If there are some contigs which don't get any BLAST hits, they are blasted again, but now using another algorithm called BLAST-X, which does the matching at the protein level. And then those results are reported as well. If there are still contigs that didn't match anything, then those contigs are reported separately, and the size distribution is shown so that you can judge which ones are potentially coming from new viruses. For this BLAST steps, there are some filtering parameters, so not all BLAST results are given or reported to you, but you can set thresholds. So, for example, you can request that the BLAST match must cover at least this much of the contig in order to be reported. So typically only contigs which match by at least 75% of their length are reported. And also the other way around, in order to report a virus reference match, you might require that at least 10% of that virus must be covered by your contigs. And you can also set parameter to require a certain minimum read depth. So then to the result files. You will get lots and lots of result files, but here I have indicated the main ones in purple. So a good starting point is a log file that VirusDetect produces, because that will tell exactly how many contigs it managed to assemble, how many viruses it detected. Then the viruses that were detected are reported in a table with a lot of supporting information in this HTML file. And then for each virus that was discovered, also a PDF file is produced. So if your sample happens to contain several viruses, then you will end up with a lot of PDF files as well. Then, like I said earlier, there is the those contigs that don't match anything by BLAST are also reported to you, as they can be potentially new viruses. So if we just have a quick look, again, you might not see this very well, but I will show you later where you can find this file to have a look. So essentially here VirusDetect reports that it started in this case with 50,000 reads. Out of those, roughly 11,000 aligned to reference virus sequence database, and the reference alignment-based assembly managed to make 62 contigs. Now then, the other assembly, the de novo assembly, was also performed, and before that the host-derived small RNAs were removed. In this case the host was potato. So reads were mapped to the potato genome, 30,000 reads out of the 50,000 mapped. So those were removed because we are not interested in host reads. And the ones that didn't map there were then kept and used for the de novo assembly, which essentially produced 39 contigs. So then the redundancies were removed, and altogether, after all that, so about 70 unique contigs were generated, which were then used to identify viruses, and in this case 2 viruses were identified by blast N and nothing with blast X. If you use the VirusDetect tool in Chipster, you also get other information in this file. It basically lists all the result files you have. Then if we look at the table listing the blast matches, it looks like this one up here. So you have the name of the viruses detected, what was the coverage, what was the length, how many contigs were supporting this, and so forth. So down here you have explanations for all these points here. And to keep in mind what you want when you look at this is a good match has typically high coverage and high depth. So like here the coverage is for the first one it's almost 89%, and for the second one 97%. And then for these, if you look at the more precise results, so let's look at the second one here. So for that you also, so for each virus you get a PDF, and that looks like this. So in blue you have the virus reference, and below in red you have the contigs that match to it. And for each contig is also shown the actual alignment. Finally, those contigs that didn't match by blast to anything are also given in a table, and those which are shown green are potentially new viruses. So the size distribution of the reads for these match what we would expect from small interfering RNAs. So here you have the different lengths, and as you can see, these guys, most of the reads for these were in this bracket, 21 to 22. There is one that wasn't, which were longer, and that is not proposed to you as a potential new virus. Then you also get a whole heap of sequence files, FASTA files, containing the contigs, containing the blast N matches, and the references, and so forth. One question that you might wonder is how many reads should you have? So when you order sequencing of your samples, how many reads do you want? Because of course the more you ask, the more you pay. Typically you get away with a quite small amount, and but of course the more reads you have, the better reference coverage you get and the longer contigs you get. So this particular image comes from the publication of VirusDetect. So here you have in black the reference virus, which is about 10,000 bases long, and then a sample that was sequenced at different levels. So there was a sample which had very, very little reads, 15,000 reads only. And as you can see, there is only one little contig made. With 30,000, you get more. And as you go higher, so this is 300,000, you are already start to get quite long contigs, covering most of the reference virus. And the more reads you can afford to have, the longer contigs you get, and you get actually pretty much perfect coverage. So the whole length of the virus is covered. Then soon we will move to the Chipster software, but I just want to show you here in slide a couple of things about it still. So in Chipster, the analysis tools are grouped into categories, and VirusDetect tools live in a category in a category called small RNA-seq. There are two tools there. One is called VirusDetect, and the other one is VirusDetect with own host genome. So the difference between these two is that so Chipster provides some host genomes for the most common organisms, for several plants, for several animals. But it could be that you are studying a more exotic organism perhaps, and Chipster doesn't have genome for that host. So in that case, you need to provide it yourself as a FASTA file. So then you would use this second tool here. So in Chipster, when you run VirusDetect, you essentially get two files. One of them is a log file, the one that I was just showing to you, and the other one is a tar file. So a tar is a package of files, actually. And the reason why we give you a tar package instead of zillions of individual files is that this allows you to actually decide yourself which ones do you want to look at without the interface getting too messy with many files. So you can extract result files from this package using a tool in the utilities category called extract tar file. And then one more thing which is important if you are studying an organism, a host for which Chipster doesn't offer the genome, and you need to give it yourself as a FASTA file. What happens when VirusDetect uses your host genome as FASTA is that it has to actually convert it from FASTA to a genome index file before it runs the PWA aligner. And this process, depending on the size of the genome, but if the host genome is big, this process can actually take several hours. So you probably don't want to spend several hours on this every time you run a different sample. So what we have done is that when you run this tool and you get the tar package, as always, in that tar package we have actually put the index file that was made during that run for your host genome, so that next time when you come to run it, you don't need to anymore give the FASTA file for your genome. You can give this ready-made index file, and that means that your analysis will run much faster. Then finally, what I wanted to say here is that if you are interested in VirusDetect but you don't have your own data yet, you can still get familiar with the system in Chipster, because we have made some sort of ready-made analysis for you that you are welcome to inspect. And this is actually something that I'm going to show you now. So I'm taking you to Chipster now. Again, this might look a bit small, but you might not be able to read all the text, but I will try to read it for you. And so what we are looking at here is I have an example session. So you can get these example sessions if you go to File, Open Example Session. It gives you a big list of ready-made sessions that allows you to get familiar with things. So in this particular session, we have data from potato samples, and we want to detect viruses there. So we have 3 different samples, and they have different amounts of reads. Now, I will just show you the sample tree, which is here, which had 250,000 reads. So it's a FASTQ file. I have selected it. Then I have gone to the tool category small RNA-seq, selected the tool VirusDetect, for which you can see the parameters here, and I have run it. And as a result, I got these two files. So the first one here is the log file that I was telling you about. So this text is very small, don't worry if you don't see it. You can log into Chipster afterwards and look at this in detail. And the other file is the tar package containing the rest of the result files. Now I have decided to extract from this package all the HTML files and all the PDF files. So essentially I have run the tool extract tar, and I have selected to get all the files which have the HTML extension, and same for the PDF extensions. And if we have a quick look at the blast result file, which is the first one here, you see that this actually looks very similar to what I had on the slide. Now, if you want to know more about VirusDetect and, for example, look at the manual page with all the things I was showing you on the slide, you get it from this More Help button. And then to finish with, I show you another case. So here we have raspberry data. Now, raspberry is a bit more exotic genome as we think. So Chipster does not have that genome readily available, but you can get it from internet. So what I have done here is I have brought it in, and that's now my host genome. I have run VirusDetect as before, and then I have extracted from my tar package again the HTML files and the PDF files, but also now a tar file. So the host genome index that was made, painfully, taking a lot of time, I extract that from the result files, and when I come to run my next sample, I can then use this index. So I don't need to go back to the FAST.genome anymore. I use the ready-made index and run my VirusDetect. So I think that's all I wanted to say now.